Imagine, if you can, a landscape with no trees. Imagine standing on a soft riverbank somewhere on the surface of the Earth 400 million years ago and looking out across a coastal plain that is green in patches, brown in patches, glistening with the slow water of a tide that never quite goes out and never quite comes in. The plants at your feet are small. They are mostly low to the ground, knee-high at the most, with simple branching stems and no leaves the way you would recognize leaves. The tallest of them, in some sheltered hollows, may rise to your waist. None of them is a tree. None of them has yet learned how to be a tree. The sky, with no canopy to filter it, falls all the way to the soil. And rising from that flat green plain, towering above everything alive, are columns. They are not stems, they are not trunks, not exactly. They are smooth, woody, slightly tapering pillars of organic tissue, anchored into the wet ground at their bases and rising in the largest specimens to perhaps 8 meters into the still air. That is more than 25 feet. That is taller than a giraffe. That is taller than a modern 4-story building. They are the tallest living things on Earth. They are not fungi exactly, though for a long time we thought they might be. They are not plants. They are not algae. They are not lichens, not in the modern sense. As of the most recent careful analysis of their fossilized cell walls and inner structure, they are nothing we have a clean name for at all. They appear to be a vanished branch of life, a lineage of multicellular organisms that never gave rise to any descendants we can find today, having ruled the early continents for tens of millions of years, simply ended. Given by a 19th century geologist who thought they were conifers, is Prototaxites. We will sit with them for a while. Over the next hour, as the light moves slowly across whatever sky is above wherever you are now, we are going to walk through what we have learned about these towers. We will go back to the Quebec field season of 1843 when a young geologist first pulled their fossil from a riverbank and gave them a wrong name. We will walk through the long century and a half of arguments about what they actually were. We will look inside the wood, the strange tubular network that nothing else before or since has ever quite produced. We will sit on the early Devonian floodplain in their shadow, and we will try gently to imagine what kind of creature this was. And then we will read the most recent science from the year 24 and the year 26 that has unsettled even the most patient assumption we had about what they were. There is no rush. The story is older than rush. The plain is quiet. The plants at your feet are not yet trees. The pillar in front of you is, in the deepest sense, alone in its kind. And the world has rarely been stranger than it was on the day this happened. Before we go further with the towers themselves, let us get the time right. Prototaxites lived during the late Silurian and early to middle Devonian periods, in a stretch of geological time that ran from approximately 430 million years ago to about 360 million years ago. That is a window of 70 million years, almost 20 times the entire age of our genus, during which these organisms were a regular feature of the wet lowlands of the continents. To set them in your mind, consider what else was true in those 70 million years. The continents were arranged in patterns you would not recognize. The largest landmass was a great southern continent called Gondwana, which contained the future Africa, South America, India, Antarctica, and Australia, all welded together. To its north were several smaller continents, including Laurussia, an early ancestor of much of modern North America and Europe. The seas between them held trilobites, brachiopods, primitive jawless fish, and the early jawed fishes that would give rise to everything that swims today. On land, things were just beginning. The very first land plants, small leafless rhiniophytes, had appeared in the late Ordovician, roughly 450 million years ago. By the early Devonian, when Prototaxites was at its peak, vascular plants had begun to develop the basic equipment they would need to grow taller: internal water-conducting tissue, lignin in their cell walls, branching axes. But they had not yet learned to put any of that to use at scale. The tallest definite vascular plants of the early Devonian were perhaps a meter high, sometimes two. Most were much smaller. Animals on land were even more tentative. The first known terrestrial animals, small arthropods, the early ancestors of millipedes, mites, and primitive insects, were creeping through the moss-thick, low vegetation. There were no four-legged vertebrates yet. There were no amphibians, no reptiles, no anything walking on bone. The landscape was wet, low, green, and quiet. And rising above it, in scattered groves and along the riverbanks of the wet lowlands, were the towers. This is the strange contrast that makes Prototaxites so unusual. They lived in a world where everything else on land was small. They were not just taller than their neighbors. They were 5, 10, sometimes 15 times taller than the next tallest organism on the same patch of ground. There has never been another time in Earth's history when one type of land organism so dramatically out-towered all the others. There may never be another such time. For 70 million years, Prototaxites was the largest living thing standing above the dry surface of the planet. And then, gradually, the forests rose and the towers vanished. But for now, in our imagined visit, the forests have not yet arrived. The plants at your feet are still small, the plain is still flat, and the columns are still here. The world without trees is hard for a modern mind to picture. Almost every land surface you have walked on in your life has had, at some scale, trees on it, somewhere. Even deserts have scattered trees in the wadis. Even tundra has bonsai-thick groves of dwarf birch. The grasslands of the Great Plains, in their original state, had cottonwoods along every river. The flat steppes of Central Asia have poplars and elms. The image of a treeless landscape, in the modern imagination, is a kind of wound, bare, exposed, not quite right. For most of the Earth's history, the wound was the default. There were no trees because there were no trees yet. The genetic and structural innovations that make a tree possible, secondary growth, deep roots, a vascular cambium that lays down new wood season after season, leaves capable of running photosynthesis at the canopy's height, none of these had been assembled. Plants on land for hundreds of millions of years were small. The first creatures with the body plans of a tree did not appear until the late Devonian, around 385 million years ago. Their leading representative was Archaeopteryx, a fern-like progymnosperm that grew, in some species, up to 30 meters tall. By the Carboniferous, 60 or 70 million years later, full forests of lepidodendron and sigillaria, the great-scale trees of the coal swamps, would dominate the wet lowlands of every continent. The Earth would be at last a wooded planet. But the towers we are walking among in the early Devonian of our imagined visit predate all of that. They predate Archaeopteryx by about 30 million years. They predate the first true tropical forests by 60 million years. They predate the first known terrestrial vertebrates by 20 million years. They predate, in the cleanest sense, almost everything we associate with terrestrial ecosystems. What this means is that Prototaxites stood in landscapes that were almost entirely open. There was no canopy to compete with for light. There were no taller organisms to shade it. The wet patches of the early Devonian floodplains, where rhyniophytes and zosterophils and trimerophytes huddled in their knee-high mats, were ground over which Prototaxites rose almost without competition for sunlight. This is one of the keys to understanding why such a strange organism could exist. Without a forest to live in, without a competing canopy of green, a structure that grew tall could simply, for a long time, be tall. Whatever Prototaxites was, whatever its tissues did, whatever the particular biology of its growth happened to be, the world handed it the sky for the taking, and it took. For 70 million years it kept taking. When the forests finally arose, the sky was no longer a free gift. The first known specimen of Prototaxites was pulled out of a Devonian sandstone in eastern Quebec in the summer of 1843. The man who found it was John William Dawson, a young geologist at the very beginning of what would become a long and distinguished career. He was 23 years old. He had been recently appointed to the Geological Survey of Canada. He was working along the Gaspé Peninsula, the long northeastern arm of Quebec that curves out into the Gulf of St. Lawrence, examining the marine and terrestrial rocks of the Gaspé sandstones. He was looking, that summer, for evidence of the early life of the Devonian period. What he found was a piece of fossilized organic material the size of a log. The piece was about 30 centimeters in diameter and roughly a meter long. Its outer surface was rough but cylindrical. When he broke a section away, showed a fine pattern of small tubular structures running along its length. There were no annual growth rings, the way you might see in a modern conifer. There were no leaves, no branches, no obvious bark. But the overall form, long, cylindrical, woody, was the form of a tree trunk. Dawson took it for a tree. This was a reasonable conclusion. The Devonian was, at the time, only just beginning to be understood as a period of early land vegetation. The expectation among geologists of the 1840s was that wherever land plants were preserved, they would resemble, in some primitive form, the trees and shrubs of the modern world. A long, woody cylinder from a Devonian sandstone in that intellectual climate was almost certainly going to be interpreted as a tree. Dawson initially set the specimen aside. He was busy with other matters: the broader geological survey work, the cataloging of the Gaspé fossil flora, the slow assembly of a regional stratigraphy. He noted the specimen and moved on. He would return to it more carefully 16 years later. In 1859, after additional specimens of similar form had been recovered from the Gaspé sandstones and from related Devonian rocks elsewhere, Dawson published a formal description of the organism. He gave it a Latin name. The name was Prototaxites. The first part, proto, meant first or earliest. The second part, taxites, was a reference to the genus Taxus, which is the modern yew tree. Prototaxites, in Dawson's mind, was an early relative of the yew. It was the oldest known coniferous tree. He was wrong in a particular way that would take more than 140 years to fully untangle. But the wrong name stuck. The wrong name is still its name today. In all those decades of additional study, given Prototaxites a better name. The name we use every time we describe these towers in the modern scientific literature still says, in its Latin roots, that this organism was an early conifer. The name is wrong. The name is, in some sense, the first ghost in the long history of trying to understand what these things actually were. Almost immediately, the Conifer hypothesis ran into trouble. When other geologists looked at the specimens Dawson had described and at additional material that came in from related Devonian sites in Europe and elsewhere, they noticed that several features did not fit. The internal structure was wrong. The wood of a conifer is composed of long parallel cells called tracheids, with characteristic pit patterns on their walls and a regular arrangement that produces the visible grain of the modern wood. The internal structure of Prototaxites was different. Its tissue was made up of small tubes, much smaller than conifer tracheids, branching and connecting in a pattern that did not match any modern tree. There were no growth rings. The cylinders were uniform along their lengths. There were no clearly identifiable roots, no clear leaf attachments, no reproductive structures of any sort that resembled cones or pollen organs. By the 1870s, alternative hypotheses had begun to circulate. Some researchers proposed that Prototaxites was actually a giant alga, perhaps a freshwater organism that had adapted to a partly emergent lifestyle along the river margins of the Devonian. The internal tubular structure was consistent, in a rough way, with certain large algae. The cylindrical form could be reinterpreted as an upright thallus rather than a trunk. The fact that Prototaxites was always found in waterlogged sediments, never in clearly upland deposits, supported the idea that it lived along watercourses. Other researchers argued for a fungal interpretation. The tubular internal structure of Prototaxites resembled in general terms the high-fall networks of large fungi. Fungi are known to grow large under the right conditions. Modern wood decay fungi can produce fruiting bodies of considerable size, and the giant honey fungus of the modern American West, Armillaria gallica, produces underground mycelial networks that, by some measures, are the largest single organisms on Earth. Still, other researchers proposed lichen interpretations. A lichen is a symbiotic association between a fungus and a photosynthetic partner, usually an alga or a cyanobacterium. Some modern lichens grow in upright forms. The argument was that Prototaxites might be an unusually large lichen, with the photosynthetic partner being some now extinct alga and the fungal partner being the structural framework. For most of the 20th century, no single hypothesis was decisively confirmed. The fossil record of Prototaxites is, by Devonian standards, reasonably good. Specimens have been recovered from many sites worldwide. In addition to the Gaspé, from Saudi Arabia, from Australia, from the Czech Republic, from various European Devonian basins, and from the famous Rhynie Chert of Scotland, where exceptional preservation in silica has frozen the cellular detail of an entire early Devonian wetland ecosystem in glassy clarity. But fossil cylinders, even well-preserved ones, can only tell so much. Without DNA, without protein analysis, without unambiguous reproductive structures, the high-level question, what kind of organism this actually was, could not be settled by morphology alone. For more than 140 years, the question stayed open. The towers stayed mysterious. The name said Conifer. The wood said something else. The interior was unlike anything alive, and the conversations went, every few decades, around again. The first major clue came from chemistry. In 2007, a team led by Kevin Boyce, then at the University of Chicago, published a paper in the journal Geology that took the question into a new domain. Rather than relying on morphology, the team used carbon isotope analysis to look at the chemical signature of Prototaxites tissue. The principle is elegant. Every photosynthetic organism, plant, alga, or photosynthetic bacterium, fractionates the carbon isotopes of its food. Plants prefer to take up the lighter carbon-12 isotope over the slightly heavier carbon-13, and they leave a measurable signature behind. The carbon-13 content of a fossil photosynthetic organism is a fingerprint of its photosynthetic chemistry. Organisms that do not photosynthesize, heterotrophs which eat their carbon rather than fixing it from the air, show a different pattern. Their carbon-13 content reflects the diet of whatever they ate, not the slow fractionation of atmospheric carbon dioxide. Their isotopic range across a population tends to be broader. Different individuals eating different sources end up with different isotopic compositions. Boyce and his team measured the carbon isotopes of multiple Prototaxite specimens from multiple Devonian sites. The result was striking. The isotopic composition of Prototaxites varied significantly from specimen to specimen, in a range much wider than would be expected for a photosynthetic organism eating atmospheric carbon dioxide. Different individuals had different signatures. Some were enriched, some were depleted. The pattern was inconsistent with photosynthesis as the primary carbon source. The pattern was, however, consistent with heterotrophy, with organisms that ate their food rather than producing it from sunlight. This suggested that Prototaxites was not a plant, it was not an alga. It did not run photosynthesis as its primary metabolism. It was, in some way, eating something, taking carbon from one source or another in its environment, building its enormous biomass out of food rather than out of sunlight. The conclusion the Boyce team drew in 2007 was that Prototaxites was probably a fungus. This was the most parsimonious explanation. Fungi are heterotrophs. Fungi are known to grow large. Fungi were known to exist in the early Devonian. The internal tubular structure of prototaxites resembled, in rough terms, the hyphal networks of modern wood decay fungi. If the carbon isotopes said heterotroph and the morphology said tubular, then the simplest reading was fungus. For about 15 years after the Boyce paper, this became the consensus. Most popular accounts of prototaxites, the magazine articles, the museum displays, the documentaries, described it as a giant prehistoric fungus. The story of mushrooms growing 60 feet tall entered the public imagination during this period, and it was based broadly on the Boyce team's interpretation. It was a clean story. It was a satisfying story. It made a difficult organism finally make sense. It was also, as the most recent work has shown, almost certainly wrong. Before we get to the most recent twist, let us spend some time inside one of the towers themselves. Most of what we know about the internal structure of prototaxites comes from a single extraordinary fossil deposit. It is called the Rhynie Chert, and it sits modestly beneath a small farm in the village of Rhynie in Aberdeenshire, Scotland. The Rhynie Chert formed about 407 million years ago, during the early Devonian, when a hot spring complex on the bank of an ancient river repeatedly flooded the surrounding wetland with silica-rich water. The water saturated the sediments, the plants, and the small organisms living in the wet ground, and as the silica crystallized in place, it preserved them, cell by cell, in glass. The Rhynie Chert is one of the most exceptional fossil deposits on Earth. The level of preservation is so fine that individual plant cells, individual fungal hyphae, individual bacterial filaments can be seen and measured under a microscope. Every Devonian wetland organism that fell into the silica was caught, frozen in mineral form, in a moment of life that has not aged since. Among the organisms preserved in the Chert is a smaller relative of the great Prototaxites of other deposits. It is called Prototaxites tate. It is named for William Edward Hoyle Tate, the Aberdonian doctor and amateur paleobotanist who first recognized the Rhynie deposit in 1912. Prototaxites tate specimens in the Chert are smaller than the great trunks of the Gaspé and Saudi Arabian sandstones, only a few centimeters in diameter, perhaps a meter or less in height, but they preserve, in glass, the cellular detail that the larger fossils have lost. When you look at a Rhynie Prototaxites under high-resolution imaging, what you see is a network of tubes. The tubes are not simple cylinders. They are of several distinct types, varying in width from a few microns to a few tens of microns. Some are thin-walled and translucent. Some are thicker-walled and pigmented. They run through the body of the organism in a complex three-dimensional weave, branching, anastomosing, that is, joining and separating again, and connecting into a tightly linked internal network. This is the structure that, for many decades, was compared to fungal hyphae, and, in a rough sense, it resembles them. Modern fungi build their bodies from networks of tubular hyphae that branch and connect in ways superficially similar to what is seen in Prototaxites. But on careful examination, the resemblance starts to break down. Modern fungal hyphae have characteristic septa, internal cross walls that divide the tube into cells. Prototaxites tubes do not have these. Modern fungal hyphae produce specific reproductive structures, spores in particular arrangements, fruiting bodies of recognizable forms. Prototaxites has produced no clearly identifiable reproductive structures despite extensive search. Modern fungal cell walls are composed of chitin, a tough nitrogen-containing polysaccharide that is one of the diagnostic markers of the fungal kingdom. The cell walls of Prototaxites, when we get to test them in the most recent work, turn out not to contain chitin at all. But we are getting ahead of the story. For now, simply imagine the tower from the inside. Imagine a slow forest of tubes, some thin and some thick, weaving through a meter-thick body, branching and connecting and growing in a pattern that is unique to this lineage. Imagine a body that is not wood, Not flesh, not familiar mushroom tissue, but something that resembles all three at different scales without quite being any of them. That is the inside of Prototaxites. We are still trying to understand what made it that way. Now let us be careful about the size. The most popular framing of Prototaxites, the framing that gives this hour its title, is that they were mushrooms that grew 60 feet tall. This is evocative, it is memorable, it is, in some museum displays and popular articles, repeated almost as a slogan. And for capturing the strangeness of a world before forests, it does its job. But the careful sizing is a little smaller than that. The largest reliable Prototaxites specimens, the ones with measured dimensions in published scientific literature, reach approximately 8 meters in height and roughly 1 meter in diameter. 8 meters is more than 26 feet. It is taller than a giraffe. It is taller than a 4-story building. It is, in any honest sense, enormous. 60 feet in metric terms is closer to 18 meters. That is more than twice the documented size of the largest Prototaxites we have found. The 60 feet claim, while popular, has no clear support in the published fossil measurements. It seems to have arisen in the late 20th century through a kind of amplification in the popular literature, each retelling adding a little more height until the figure landed somewhere safely impressive but unsupported by the actual specimens. We will not use it again in this hour to describe what these organisms actually were. What they actually were, on average, was probably more like a few meters tall, perhaps 2 or 3 for the typical mature specimen, with the largest reaching the 8-meter range. Their diameter was substantial. A Prototaxite's trunk, viewed end-on, would have been about 1 meter across in the largest cases, the diameter of a modern oak trunk, though without the bark and without the rings. Imagine then a column the diameter of a large oak, 3 meters tall in most specimens, 8 meters tall in the giants. Now imagine that column standing alone in a landscape where everything else is below your knees. This is the actual scale at which Prototaxites was extraordinary. Not the absolute size, which by modern tree standards is modest, but the size relative to its neighbors. A Prototaxites in the early Devonian floodplain was 3 to 10 times taller than anything else within sight. A walker through the Devonian wetlands, though no animal walking through a Devonian wetland would have been very large either, would have moved among ankle-high mosses and waist-high vascular plants, and would have looked up occasionally at something that towered above the entire landscape. The towers were not record breakers in absolute terms. They were the tallest thing in their world. That is a different kind of strangeness, and arguably a more interesting one. So what did they live on? If Prototaxites was a heterotroph, and the carbon isotope evidence makes this strongly likely, regardless of what it ultimately turns out to be in higher-level taxonomy, then it was eating something. The question is what? The early Devonian wetlands were rich in organic matter, even if they were not yet rich in trees. The land plants that did exist, small, leafless, often wet-loving, produced fallen tissue, decaying litter, dead stems and roots that accumulated in the soft sediments along watercourses. Microbial mats, algae, cyanobacteria, and the early fungi all thrived in the same wet environments. Fish carcasses washed up from the rivers, dead arthropods accumulated in the leaf litter. There was, in other words, a rich pool of organic carbon for any heterotroph capable of accessing it. Modern wood decay fungi access this kind of resource by breaking down the tough structural polymers of plant tissue, primarily cellulose and lignin, using extracellular enzymes that they secrete into the substrate. The breakdown products are then absorbed by the hyphae and used to build new fungal tissue. This is how a modern shelf fungus on a fallen log feeds itself. It is also how the giant Armillaria mycelial networks of the modern Pacific Northwest spread, slowly, decay-feeding, through the vast underground litter of forest floors. Prototaxites may have done something similar. The Boyce team's 2007 analysis suggested that the organism was breaking down a mix of substrates, drawing carbon from multiple sources of decaying plant material in its environment. This is consistent with a saprotrophic, that is, decomposer, lifestyle. It would explain why Prototaxites is found preferentially in waterlogged sediments where organic matter accumulated. It would explain why the organism could grow so large despite the absence of leaves and photosynthesis. There was no upper limit on its biomass except the supply of decaying material. Other possibilities have been proposed. Prototaxites might have been an endosymbiotic organism, hosting photosynthetic algae or cyanobacteria in some way and benefiting from their carbon fixation indirectly. It might have been a complex lichen with a structural body and embedded photosynthetic partners. It might have done some combination of things, primary heterotrophy supplemented by symbiotic photosynthesis, or vice versa. The honest answer is that we do not know with certainty. What we can say is that whatever Prototaxites was eating, it was eating effectively enough to grow into a column 3 meters wide and 8 meters tall in a world where almost nothing else exceeded a meter. It was, in its niche, extraordinarily successful. It dominated the early Devonian terrestrial landscape for tens of millions of years. It outlasted the trilobite radiations of the Silurian. It outlasted multiple episodes of climate change. It persisted through the rise of jawed fish, the diversification of early vascular plants, and the first faint stirrings of vertebrate life on land. Whatever its diet, the diet worked. For 70 million years, the diet worked. And then, when something changed in the world that supported it, the diet stopped working, and the organism, having had no descendants we can trace, simply ended. We will get to that ending in a moment. But first, the most recent twist. In the spring of 2026, the journal Science Advances published a paper that quietly upended much of the consensus that had built up around Prototaxites in the previous 2 decades. The paper was led by Corentin Laurent, a postdoctoral researcher at the University of Edinburgh, working with colleagues from Edinburgh and from the Diamond Light Source synchrotron in southern England. The team had set out to test, in the most rigorous chemical and structural detail then available, the proposition that Prototaxites was a fungus. They focused on Prototaxites tighti, the small Rhynie Chert relative, because the Rhynie Chert preserves cellular detail at a level no other Prototaxites deposit can match. They used synchrotron infrared spectroscopy to analyze the molecular composition of the cell walls. They used 3-dimensional X-ray imaging to map, at sub-micron resolution, the internal tubular network. They compared the results to a library of known fungi, both modern and fossil, and to other early land organisms preserved in the same chert. The results, when the team assembled them, did not match the fungal hypothesis. 2 key findings stood out. First, the cell walls of Prototaxites tighti contained, prominently, polymers most similar to fossilized lignin or lignin-like phenolic compounds. Lignin is a structural polymer characteristic of vascular plants. It is what gives wood its rigidity. It is one of the most diagnostic chemical signatures of plant tissue in the fossil record. The cell walls of Prototaxites did not, in any detectable amount, contain chitin or chitosan. These are the structural polymers that define the fungal cell wall. They are present in every modern fungus that has ever been tested, and in every fossil fungus where chemistry can be measured. Their absence in Prototaxites is, by current standards, very strong evidence that Prototaxites was not a fungus. The 3-dimensional structure of the Prototaxites tubular network turned out to be unique. The team used the synchrotron imaging to map the precise geometry of the branching tubes. They compared the geometry to that of fungal mycelia, of plant vascular tissue, of algal thalli, of every comparable structural pattern in the modern biological literature. The Prototaxites pattern matched none of them exactly. The branching angles, the connection density, the spatial arrangement of the different tube types, All of these were unlike anything known from any modern organism, fungal or otherwise. The combination of plant-like cell wall chemistry and unique structural geometry suggested that Prototaxites was neither a plant nor a fungus in the standard sense. It was something else. The Loron team's interpretation, published in early 2026, was that Prototaxites represents an extinct lineage of multicellular eukaryotes, a branch of complex life on land that arose somewhere in the late Silurian, dominated terrestrial ecosystems for tens of millions of years, and then disappeared without leaving any modern descendants we can identify. In the framing that quickly spread through the press coverage of the paper, Prototaxites became a lost kingdom of life. The phrase is dramatic, and the underlying biology is genuinely uncertain. We do not know whether Prototaxites belongs to its own kingdom, its own phylum, or some narrower category, but the basic point holds. Whatever Prototaxites was, it was not part of any major group of organisms still living today. Its closest living relatives, if it has any at all, are unknown. The first organism to grow tall on land may have been a kind of life that no longer exists. The thought is worth sitting with, slowly. For most of the history of multicellular life on Earth, we have assumed, for good reasons, that the major lineages we know today represent more or less all the major lineages there have ever been. Animals, plants, fungi, and the various groups of protists. With time, we have added complexities, the rise of new lineages within these groups, the extinction of subgroups within them, the gradual elaboration of the family tree. But the broad framework, animals, plants, fungi, has stayed mostly intact. What the Prototaxites finding suggests is that this framework is incomplete. There may have been entire lineages of complex multicellular life, lineages with the size and ecological dominance to be the largest organisms in their world for tens of millions of years, that arose, persisted, and then vanished, leaving no trace in the modern biota. The Prototaxites lineage, on the current evidence, was one such. There may have been others. The implications, if you sit with them, are gentle but unsettling. It means that the diversity of complex life on Earth, taken across all of geological time, is wider than the diversity we currently observe. It means that the modern world, with its plants and fungi and animals, is not the full inventory of what was possible. It is, in some sense, a sample. The actual range of multicellular body plans, of metabolisms, of ecological strategies that complex eukaryotic life has explored is broader than what we can study today. Range is preserved in the fossil record, in lineages we've always assumed must fit somewhere into our existing tree. Prototaxites for 140 years was assumed to fit. It was assigned to plants, then to algae, then to fungi, then back again. The assumption was that it must belong somewhere in the modern tree. We just had not yet figured out where. The most recent science is suggesting gently that perhaps it does not. Perhaps it belongs to nothing in the modern tree. Perhaps it belongs to a branch that has fallen entirely. There is no clean modern parallel for what this means. We do not, in any other branch of biology, have lineages this large that have left no descendants. The dinosaurs left descendants, birds. The trilobites, by some interpretations, left distant arthropod descendants. The strange creatures of the Ediacaran biota are still debated, but at least some of them have plausible relatives among modern animals. Prototaxites, if the Loron team's interpretation holds, has no living relative anyone has yet identified. Its kind ended at the end of the Devonian, and the family tree of life simply has a stub where it used to be. The forest of life is wider than the forest of life we now see. This is, perhaps, the deepest lesson of an organism we are still trying to understand. Now, slowly, the question of how it ended. Having dominated the early Devonian terrestrial landscape for tens of millions of years, gradually disappeared from the fossil record during the late Devonian. The most recent, reliably identified specimens come from rocks dated to around 360 million years ago, near the end of the period. By the time the Carboniferous opens in the early 350s million year ago range, the towers are gone. The fossil record does not show their decline as a sharp event. It shows it as a slow thinning. What was happening in the world around them during their decline is, by contrast, quite well understood. The forests were rising. Across the Middle and Late Devonian, plant life on land underwent the most dramatic structural revolution in its history. The genetic and developmental innovations that make a tree possible, secondary growth, the vascular cambium, deep root systems, true leaves capable of running photosynthesis at heights, were assembled in different lineages over the course of perhaps 30 million years. By around 385 million years ago, the first plants with the body plan of a tree had appeared. Archaeopteris, a fern-like progymnosperm, was leading the radiation. Other lineages, early gymnosperms, primitive lycopsids, were following. By 370 million years ago, the first true forests existed. Tall, branching leafy plants formed canopies that shaded the ground. The simple wet floodplain that Prototaxites had known was being progressively replaced by closed canopy woodland. Light, which had previously fallen freely to the ground, was now being intercepted 20 meters up. For Prototaxites, this was the end of the world it had been built for. We do not know exactly how Prototaxites was affected by the rise of the forests. If it was a heterotroph that lived on decaying material from the ground level, and the carbon isotope evidence supports this, then the appearance of true trees would have changed both its food supply and its competitive environment. The new forests would have produced enormous quantities of new organic matter, but they would also have hosted a new generation of decomposer organisms with chitinous cell walls and chitinase enzymes well suited to processing wood that could exploit the resource more efficiently than Prototaxites apparently could. If Prototaxites had any photosynthetic component to its biology, symbiotic algae, internal cyanobacteria, anything that contributed sunlight-derived carbon, the rise of canopy-forming trees would have shaded those partners out. The light that had reached the ground freely for tens of millions of years was now being absorbed at a height the towers in their slow growth pattern may not have been able to match. There may also have been competition for water, for nutrients, for soil structure. The deep roots of the new trees changed the chemistry of soils. The accumulating litter of forest floors changed the microbial communities that prototaxites had to live alongside. Whatever the specific mechanism, the towers thinned. They thinned across the late Devonian. They became less common in the fossil record. Their typical size in the youngest specimens may have been smaller than in the older ones, though the data is sparse enough that this trend is debated. By the close of the Devonian, around 359 million years ago, the lineage was gone. The end is gentle in a way. There is no mass extinction event that targets Prototaxites specifically. There is no catastrophe in the rocks. There is just the slow disappearance of a way of being on land that was no longer competitive. The forests inherited the world the towers had ruled. The towers did not return. The fossils, meanwhile, are still here. Prototaxites specimens have been recovered from a remarkable range of sites worldwide. Beyond the Gaspé Peninsula of Quebec, where Dawson first named them, they have been found in the Rhynie Chert of Scotland, the Old Red Sandstone of Wales, the Devonian sediments of Australia, the late Silurian deposits of Saudi Arabia, the early Devonian rocks of the Czech Republic, and various scattered occurrences across the Devonian basins of North America, Europe, North Africa, and parts of Asia. This wide distribution tells us that Prototaxites was not a regional anomaly. It was a global organism. Wherever the early Devonian wetlands were preserved well enough to capture organic remains, the towers, in some form, are usually there. The largest specimens have come from the Gaspé and Saudi Arabian deposits, where unusually deep burial in waterlogged sediment preserved entire trunks at near original size. The most chemically informative specimens have come from the Rhynie Chert, where the silica preservation has captured cellular detail at submicron resolution. The most numerous specimens have come from the Welsh and English Old Red Sandstone, where Devonian beds outcrop widely, and amateur and professional collectors have been finding fragments for over 150 years. If you wanted to see one in person today, you could. Many natural history museums have specimens on display, particularly in Canada, the United Kingdom, and Germany. The Royal Ontario Museum, the Natural History Museum in London, the Senckenbers Natural History Museum in Frankfurt, and various smaller institutions all hold material. The trunks themselves are unprepossessing in appearance, long brown or gray cylinders of fossilized organic tissue, often broken into segments, with no obvious feature that announces their strangeness to the casual eye. You would walk past one without a second thought, unless somebody was there to tell you what it was. That is the quiet way of these objects. They do not advertise themselves. They sit in their cases or on their shelves, named with their wrong Latin name from 1859, and they wait, patiently, for the next round of microscopy and chemistry to take another measurement of them. Each round of measurement, in the long history of looking, has changed the story of what they were. The Conifer hypothesis fell. The algae hypothesis fell. The lichen hypothesis is still alive in some quarters but weak. The fungal hypothesis, which dominated for 15 years, has now fallen too. What remains is something stranger and more humble, the recognition that we have in our museums the fossilized remains of an organism whose nearest living relative is unknown. The towers are still talking to us. It just turns out we have been mishearing them for 183 years. There is something gentle and slightly melancholy about a lineage that left no descendants. Almost every other major group of complex multicellular life that we know about, from the Cambrian to the present, has at least some echo in the modern world. The trilobites have, in their broadest evolutionary sense, distant arthropod cousins. The dinosaurs have birds. The Ediacaran biota has at least some plausible relatives among modern animals, even if the connections are debated. The strange Burgess Shale creatures of the early Cambrian, Anomalocaris, Hallucigenia, Opabinia, were once thought to belong to entirely separate phyla, but most of them have since been folded into the broader arthropod tree. The forest of life is, in this sense, mostly continuous. The branches alive today reach back, by visible threads of descent, to most of the major branches that have ever existed. Prototaxites, on the current evidence, is an exception. If the Loron team's interpretation holds, and it may yet be revised as all interpretations may be, then Prototaxites represents something rare in the fossil record. It is a major, geographically widespread, ecologically dominant lineage of complex multicellular life that arose, persisted for tens of millions of years, and then ended without leaving any modern descendants we have been able to identify. The branch grew, the branch held, the branch fell, and nothing on the current tree carries the line forward. This is, in a soft way, a lesson about the limits of inheritance. We are accustomed to thinking of evolution as a continuous unfolding, in which the diversity of life at any given moment is the product of all the diversity that came before. New lineages arise from old lineages. Old lineages give birth to new ones. The pattern, on the broadest scale, is one of expansion and connection. Fewer kinds of life in the deep past, more kinds today, with the lines of descent from then to now mostly traceable. Prototaxites complicates this picture. It says gently that some lineages do not carry forward. Some kinds of life arise, flourish, and end, with no continuing thread that connects their world to ours. The diversity of life on Earth, taken across all of geological time, includes lineages that have no echoes in the present. The modern world is not the sum of all the past. It is the surviving subset. The towers were one of the unechoed. Whatever they were doing, whatever solution to terrestrial existence they had assembled, whatever way of building a body, whatever way of feeding, whatever way of reproducing, that solution did not pass forward. It ended in the late Devonian. The genetic instructions for being a prototaxites are gone. The lineage is silent. If you wanted to bring one back, even with the most ambitious imaginable molecular biology, you could not. The DNA does not exist. The cell wall chemistry can be inferred from fossils, but the living biology that produced it, the metabolism, the development, the reproductive cycle, is unrecoverable. The towers are, in the deepest sense, finished. It is possible in a quiet way to grieve them. Not as you would grieve a person, of course, not even as you would grieve a more recently extinct species, a passenger pigeon, a thylacine, a great auk, for whom photographs and museum specimens still preserve some last echo of how the body actually moved. The grief for Prototaxites is older and gentler than that. It is the grief, if grief is even the word, of the simple recognition that the world has done some of its best work in lineages that did not survive to tell us about it. The towers were tall, the towers were strange, the towers are gone, and the modern world we have inherited, beautiful as it is, full as it is of the visible life of forests and reefs and grasslands and coral and birds, is missing them in ways we mostly do not know to miss. So we have walked slowly through what is known and not known about Prototaxites. We have stood in our imagined visit on the riverbanks of the early Devonian and looked up at the towers in their original setting. We have followed the long history of trying to name them. Dawson's Conifer in 1859, the algal hypotheses of the late 19th century, the lichen and fungal interpretations of the 20th, the carbon isotope work of the early 21st, and the most recent chemical and structural analysis that has, gently, dissolved the fungal consensus and replaced it with a question. We have looked inside the trunks, at the unique tubular network that no living organism quite reproduces. We have considered what they ate and what they might have lived alongside, and how they ended as the rise of the forests slowly closed the world they had been built for. We have sat with the recognition that the lineage left no descendants, and that we are looking, in our museum cases, at the fossils of a kind of life that is finished. There is no clean conclusion. The science is still moving. Next year, or in 5 years, or in 20, somebody may publish another paper that revises the interpretation again. The Prototaxite story has been revised on roughly 30-year cycles throughout its entire history. There is no reason to think the most recent revision is the final one. What does seem stable across all the revisions is the strangeness itself, a column 3 meters wide, 8 meters tall, made of fine tubular tissue with lignin-like cell walls but no chitin, anchored in the wet ground of an early Devonian floodplain, drawing carbon from sources we are still working out, towering over the small green plants of a world that did not yet have trees, persisting as the dominant land organism of its planet for 70 million years before the rise of true forests pushed it out. This is what was here in the early Devonian, and it was here for a very long time. If you have stayed with this voice all the way through, thank you for the company. If you have drifted somewhere along the way, thank you for that too. The towers are patient. The story is patient. The Devonian is not in any hurry, and neither is the slow accumulation of what we now think we know about what was once the largest living thing standing above the dry surface of the earth. Outside, wherever you are, the forests are tall. The trees that fill modern landscapes are descendants of plants that arose 300 million years after Prototaxites disappeared. The world has rebuilt itself with green canopies and birds and the long shadows that only trees can cast. The towers are not part of that rebuilding. They are not part of the forest you can step into tomorrow. But somewhere in the rocks of Quebec and the rocks of Scotland and the rocks of Saudi Arabia and Wales and Australia, the trunks of Prototaxites are still preserved, patient, quiet, waiting for the next round of careful science to ask them, again, what they were. Sleep well, if you are sleeping. The Devonian is far away. The forests have grown back in different forms, and somewhere in the long file of what life has tried, the towers are still standing, even if only in stone.